Hello everyone, my name is Snehlata. I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today, we will continue with the next lecture of the chapter Photosynthesis in Higher Plants. So, till last lecture, we have completed almost everything about the photosynthesis reactions, that is, light reaction and dark reaction. We saw that in light reactions, what are different cycles or different type of photophosphorylation that takes place. And then in dark reaction, we saw that based on the plant, there are different type of dark reaction that takes place, such as C3, C4, and CAM cycle. Now, today we'll discuss the last topic of this chapter, that is factors affecting photosynthesis. So, in the factors that affect the photosynthesis means that these are the factors that affect the rate or speed of photosynthesis. They affect rate or speed. Okay. So, these factors are of two types. The first one is the internal factor, and the second is the external factors. Now, what do you mean by internal factors? So, the internal factors are basically plant factors. So, these are the factors that are present within the plant and that depends on the genetic composition of those plants. Okay. So, these factors are the internal factors are such as properties of the leaves, such as age, size, area, and orientation of leaf. Orientation means with respect to light. So, orientation of leaf. So, these are the properties that these are the properties of the leaf that can affect the rate of photosynthesis. Secondly, number of chloroplast that is present in a leaf. So number of chloroplast that is present in a leaf, because we know that this entire photosynthesis that takes place in the chloroplast. So its number also this affects the rate. Then third is number of mesophyll cells. Now, because in most of the cases the dark reaction takes place in the mesophyll cells, right? Or in some cases it takes place in the combination, like in C4 cycle. But the mesophyll cells are important, and in most of the time, this ribisco enzyme that catalyzes the dark reaction is present in the mesophyll cells. Hence, the number of these mesophyll cells are also important. Then the next is chlorophyll content, because by now we have understood why chlorophyll content is important. Because this chlorophyll is what? This is it forms the reaction center and the antenna, right? From where this light reaction starts. So the chlorophyll content is important. And we have also seen by an experiment that if a leaf do not have chlorophyll, that was this variegated leaf experiment. So the photosynthesis does not take place there, right? So the chlorophyll content is also important. Then internal CO2 concentration, internal carbon dioxide concentration that is present in the leaf. So internal carbon dioxide concentration in leaves. So it does not matter how much carbon dioxide is present outside, right? But it should reach inside the cell in the leaves, right? If it does not happen, then the photosynthesis won't take place, right? And we have also seen prove this thing by an experiment in our first lecture, in which the KOH that was used to absorb the CO2, in that case, even you provide the CO2 from outside, but if it cannot get absorbed by the leaves, it will have no effect, and hence the photosynthesis won't take place, right? Then the next factor is number of stomata. So the stomata, we know these are bean-shaped structures present in the leaf mesophyll cells in the leaves. So what they do, they undergo opening and closing, and hence allow the exchange of gases and water through it, right? So the number of stomata also play important role. If the number of stomata is very low, then exchange of gases like CO2 won't pass, the water won't pass. Or if the number of stomata is very high, in that case, what happens? There will be excessive water loss, right? So Hence, the number of stomata, it also affects the photosynthesis. Now, so these are the internal factors. And if in one word we have to write that how these internal factors or what govern these internal factors that in turn affect the photosynthesis. So these internal factors depends on genetic predisposition. It depends on genetic predisposition. So it means, what does genetic predisposition means? It means what are the genes or what is the genetic constituent of that plant. So we know that all the living organisms, they consist of, they have certain genetic composition, right? And this genetic composition governs all the reaction that take place in our body or in any living thing. So this photosynthesis also, so all these internal factors, they are regulated by, you can say they depends on genetic predisposition, or they are regulated by genes. They are regulated by plant genes. Okay, so these are the internal factors. Okay, now what are the external factors? So external factors, these are what extrinsic factors, right? External factors are also called as extrinsic factors. That is, these things will be provided from outside. It does not depend on the genetic composition of the plant, but it is dependent on the environment. So external factors. So governed by it is governed by environment, right? It is governed by environment. Okay. So, and among this, the four important external factors are, the first one is light. The second is CO2. Third is temperature. And fourth is water. So these are the four important external factors that we'll study in detail. Okay. Okay. So before moving to those external factors, let's talk about one law. And this law is known as law of limiting factors. Okay. So what is law of limiting factor? So this law it was given by a scientist named F.F. Blackman. It was given by F.F. Blackman in year 1907. In 1907. Okay. Now, what does this law states? So it states that suppose a biochemical reaction, let's say photosynthesis. Okay. So biochemical reaction such as photosynthesis, it is governed by multiple factors that we know, right? So it is governed by internal factors that we have mentioned in the last slide. So it has internal factors. It is governed by light. It is governed by temperature, right? And it is governed by CO2, so on. So now, if there are multiple factors, then which of this factor will actually define the rate of that photosynthesis, say, or that biochemical reaction? So turns out that this law that was given by F.F. Blackman, he showed that out of all these factors, the factor which is lowest in concentration or is present in the least amount actually governs the rate of the photosynthesis or actually governs the rate of any biochemical reaction. Okay. So what is the law of limiting factor? So it is what that if a biochemical process is controlled by many factors, is controlled by multiple factors. Okay. So if a biochemical reaction is controlled by many factors, then in this case, what will actually uh, tell the rate of that reaction? So in this case, the rate depends upon the factor which is most limiting, which is most limiting. And by most limiting, what I mean, which is lowest in concentration. Okay. which is lowest in concentration, or you can say amount, or amount. Okay. Now, but this does not mean that, suppose here we are talking about carbon dioxide, we are talking about temperature and light, but we know all these are different units, right? So in this case, how we can say that which is the least in concentration? So it means that, suppose there is one term known as optimum, right? So optimum is that concentration at which you will obtain maximum rate or maximum growth, right? So suppose for light, the maximum or the optimum value is say 20, right? For temperature, for carbon dioxide, say optimum is say 10. Now, if you see that now you have kept the plant in a condition where it is receiving a light of unit 5 and it is receiving CO2 also of unit 5. Now, in this case, which will be the limiting factor? So, in this case, the rate of the photosynthesis will be governed by carbon dioxide. Now, you can ask that in both of the cases, the unit is 5, like, so it has also 5 unit, it is also 5 unit. But here we are seeing the relative. Okay, so in the case of light, if you see the optimum is 20, but it is getting only 5 units, which means it is deficit of 15 units, right? While in the case of carbon dioxide, what is happening? The optimum is 10, right? Optimum is 10 and we are getting 5. So we are deficit of, or the plant is deficit of 5 units only. It is deficit of 5 units only. Okay. So here the most, sorry, yeah, it is deficit of 5 and here it is deficit of 15 units. So the light will be the rate limiting factor. Okay. Because there is highest efficiency of the light. Okay. So in this case, light will be the your rate limiting factor. Okay. So this is how it works. So if we talk about a limiting factor, we see it in the terms of the optimum concentration first, that what is actually required and how much it is getting. And based on that, we decide the limiting factors. Now, so this is the law of limiting factor. Here, if a biochemical process, so in this term, in the term of biosynthesis, this photosynthesis, the biochemical process is what? It is photosynthesis. So the photosynthesis is controlled by multiple factors that we see, and the rate depends on the factor which is most limiting. If we talk about the example, if someone asks that if you have a low light intensity, so in the case of low light intensity, if you increase the temperature, okay, if you increase the temperature, now if someone asks that what will happen to photosynthesis, so it is saying that your optimum light intensity is low, your light intensity is less than the optimum light intensity, but you are increasing the temperature. Then will your photosynthesis increase or decrease, or there will be no change? So in this case, if there is low light intensity, no matter how much temperature you will increase, the photosynthesis will not increase, right? So there will be no change. No change in rate, right? No change in rate of photosynthesis. Because in this case, this low light is your limiting factor. It is your limiting factor. So the photosynthesis will increase only when you will increase the light intensity. So when you will increase this limiting factor, so photosynthesis will increase when we increase limiting factor. When we increase limiting factor. Okay. So no matter how much temperature you are increasing in this condition, but your temperature is not a limiting factor. It has already stated that the plant is in low light intensity. So you have to increase the light intensity in order to increase the rate of photosynthesis. So this is about the law of limiting factor. Okay. Now we have in previous slide what we saw that the external factors, there are 4 external factors that are important and they regulate the photosynthesis. Now we'll see them one by one. So the first factor is light. Now, in the light, if you talk about light, so in light we have three parameters. The first parameter is about the quality of the light. The first parameter is quality of the light. Okay. Now, what do we mean by quality? The quality means that what wavelength of light we are receiving. So it means wavelength. You can also say quality wavelength of light. Okay. Now we know that plant responds to this photosynthetically active radiations range. Okay. So plant responds responds to photosynthetically active radiations. Active radiations. Okay. These are also known as PARs. And what is this photosynthetically active radiation? It comes in the range of 400 to 700 nanometers. Okay. So the plant responds to this. And out of this, we know that T.W. Eagleman, he proved that which out of this, which wavelength is most appropriate or most optimum for the photosynthesis. So we know that the maximum photosynthesis takes place, maximum photosynthesis, it happens in the range of red and blue wavelength light. Red and blue wavelength light, right? Red and blue wavelength light. And the minimum photosynthesis, it happens in the green wavelength. Minimum photosynthesis, it happens in the green wavelength, right? So this is the quality. This is what we know about the quality of the light. Okay. So what quality of light it is receiving, based on that, it will determine the amount of photosynthesis. Okay. Not rate, but amount. Okay. So these terms are important. Amount means how much molecules are forming, because the ultimate goal is formation of glucose molecule that converts into the starch. So the quality, it determines the amount of photosynthesis. Okay. So how much carbon glucose is forming, how much oxygen is getting released, these things. But speed, speed is different from amount or quantity, right? Now the second factor or second parameter of light is what? It's, you can say, duration. Duration. So in a day, how much light a plant is receiving, right? So duration, you can say that amount of light or quantity of light received by plant in a day. Received in a day. Okay, so this is the duration, and this is also known as photo period. It is also known as photo period. Like photo means light, so light period. So during limit, for how many time the light is getting received by the plant. This is the same term. Again, this duration, it does not control the rate. Okay, so it does not control the speed of photosynthesis. Does not control the rate of photosynthesis, but it regulate the overall process. But regulate overall process. Okay. So what I mean by this, so suppose you are getting light, a good amount of light during afternoon, but in morning and evening you are not getting any light. So it will not affect the rate of photosynthesis, because rate is what? Rate is what is the how much substrate is getting product is getting formed per minute, right, or per unit time. So that thing it won't affect, but the overall amount that will be formed, that will be affected, right? So if you are getting a light during afternoon only, not in the morning and the evening, so the photosynthesis will happen in the afternoon only, so you will get less amount of product, right? But the speed will be same even if it is happening in the afternoon, right? So this is what I mean. Now, so this is the second factor. Now, what is the third factor? The third factor is the intensity of the light. The third factor is intensity. And this intensity, it actually does what? regulate your rate of photosynthesis. So these two, they regulate the amount, but intensity, it controls the rate of photosynthesis. Okay, so it controls the rate of photosynthesis. So what intensity of light uh, plant is receiving, that will ultimately decide that at what speed the photosynthesis will take place. Okay, so these two, they are deciding the amount of photosynthesis, and this intensity, it decides the rate of photosynthesis. Okay, now if we draw the curve of light intensity versus light intensity versus rate, then how the curve will look like. Okay, so if you see, now we have said, I said in the previous slide that light is intensity is affecting the photosynthesis. So does it mean that if you increase the light, infinitely, so will the photosynthesis rate will keep on increasing? The answer is no. So if you plot a graph between light intensity and rate of photosynthesis, the graph will look something like this. The graph will look something like this. Okay. So now here we know that it's not like if you even increase a light intensity by 1 unit, the plant will start doing photosynthesis. It does not happen like that. There should be a minimum amount to start. Okay. So it will never start, like the rate will never start from here. It will start something from here. But this is a say point A. Okay. This is a O point. So once the light intensity will reach here, then only photosynthesis will start increasing. So graph will should look something like this. Okay. But in NCRT, it is given from here. So now let's see what is happening. So see from here to this point, if you see from say this is B point, now this is C point, and this is not increasing, this is something like linear. Okay. Now what we are seeing three different zones. The first one is like this A point, then from here, say here to this B point, then B to C, and then C onwards. Okay. So this is one, this is second, and this is a third. Now let's see what is happening. So the first point is A point here. So A point is known as compensation point. A is what? It is compensation point. Okay. Now what do I mean by compensation point? So at this point, nothing is actually happening. Like here, the rate of photosynthesis, the net gain is nothing. Okay. Because here, the rate of photosynthesis equals to rate of respiration. So at this point, rate of photosynthesis is equals to rate of respiration. Okay. So here, there is no net gain in terms of, if you say photosynthesis is making certain product. So there is no net gain. Okay. Because whatever is being synthesized, it is getting utilized. Okay. So this is the compensation point. Now, second is what we are seeing. We are seeing that there is this this thing, right? This B point, and then there is C point. Okay. And from C point, if you see, you have achieved the maximum rate, because here after this thing has saturated. So this is your maximum rate, right? Now, if we talk about this point, this up to B. So here what we are seeing the linear relationship between the light intensity and rate, right? So here what we are seeing linear relationship, linear relationships. If I say it as A dash, this point A dash. So here A dash to B, what we are seeing linear relationship between light intensity and light intensity and rate, right? So this is the one point. So it has to be, we are seeing linear relationship. Now, when this linear relationship is happening, under what light conditions? So if you see in this condition, the light intensity is low. So this linear relationship, it occurs when there is low light intensity. Low light intensity. Okay. If there is low light intensity, there will be a linear relationship between light intensity and rate, meaning as you increase the light intensity, the rate of photosynthesis will also increase. Right? Now you have second condition where we can see that here from B dash to C. B dash to C. So here what happened? Here a kind of plateau is being now attained by the plot, right? So here the rate is becoming constant. Rate constant. So not only B dash to C, but onwards also. So they will become constant. So in this thing, what will happen? Saturation will be attained. Saturation will be attained. Now, even if you increase the intensity of the light, you will not observe or find any increase in the rate of the photosynthesis. So in this case, now light will no longer be a limiting factor. Here, other factors will become limiting. So here, other parameters will become limiting factor. Okay. So what I'm trying to say is, if you plot a light intensity versus rate of photosynthesis plot, you will observe a plot like this. Okay. In which this A point is what? It is a compensation point where the rate of photosynthesis is equal to rate of respiration. After that, when as we increase the rate of the light intensity, as we increase the light intensity, the rate of photosynthesis will increase, and it will show a linear relationship between light intensity and rate. But this situation, this linear relationship will be till the time when there is only low light intensity, right? Meaning this light intensity is the limiting factor. So in the case of low light intensity, light is what? Limiting factor. Light is limiting factor. Now, once it will receive the maximum, or you can say optimum light intensity, then it will saturate. Okay, so it will saturate, and in this case, light will no longer be a limiting factor, but in this case, other parameters will become limiting factors, say temperature, carbon dioxide, anything, depending on the situation. Okay, now we if what is mentioned is that only 10% of light, so whatever light falls onto the earth, out of this, only 10% of the light is sufficient for the plant. Okay. So only 10% of light is sufficient for plants. Now, if only 10% of light is sufficient for plants, it means that light is rarely a limiting factor, right? Because whatever light the earth is receiving or the plants are receiving, only 10% of that light is sufficient to do photosynthesis. So light will be barely a limiting factor, right? So light is really a limiting factor. So out of the four factors that we studied, it is really a limiting factor. But in certain situations, light can act as a limiting factor. For example, so in conditions like plant growing under canopy or shady region. Okay, plants growing under shades. For example, you have a garden. You have large number of these trees. So if a plant is growing below this, here it will not receive light, right? So for these plants, light will be limiting factor, right? So in conditions like plant growing under shades, light is light can be limiting factor. Light can be limiting factor. Okay. Secondly, so but we have seen that in some cases, some plant they are indoor plants, right? They do not require light. So what happens in those plants? For example, some, if you say some plants can grow under shades, right? Some plants can grow under shades. So what happens to these plants? These plants have high chloroplast number. They have high number of chloroplast. So even a small amount of light intensity, they can utilize it for the conversion into energy and ultimately glucose. Okay. So those plants that grow under shades, they have high number of chloroplast. And in most of the cases, light is not a limiting factor, except for the case where plant is growing under shades. So this is about the light, and we have seen that how light affect the rate of photosynthesis. Okay. Now, next we will move to the second factor, that is carbon dioxide. This carbon dioxide, it is the most important limiting factor. Okay. So this is most important limiting factor. Now, what does this, how much, if you talk about the atmosphere, how much carbon dioxide do we have in the atmosphere? So in atmosphere, the amount of carbon dioxide is 0.038%, or you can say 380 parts per million, or 380 microliter. Okay. So this is the concentration of carbon dioxide in the atmosphere. Now, in plants, the plants can tolerate maximum CO2 of maximum carbon dioxide concentration of 550 ppm, 550 microliter, or 550 ppm. Okay. So this is the maximum tolerance level. So under normal conditions, like in our environment, plant do not have any problem with the carbon dioxide, right? Because the carbon dioxide in the atmosphere is only 380 ppm, and plant can tolerate up to 550 ppm. Okay. Now we have studied about the different type of plants. For example, say C3 plant, right? And we have seen C4 plants. Now these plants, they have different carbon dioxide optimum. Okay. So optimum concentration of carbon dioxide that these plants requires, optimum concentration of CO2 that these plants require. So the C3 plants, they require 450 ppm, or what, 450 microliter. So ppm and microliter is same. Okay. And the C4 plants, they require 360 ppm. So if you draw a graph, for CO2 concentration and rate of photosynthesis, so we will see that this C4 plants, and if you draw, so this is your C3 plant. So after this, it will receive the saturation. And then if you will draw another graph that will look something like this. Okay. Now out of this, which will be the C3 and which will be the C4? So the C3 will be the one that has high optimum concentration, right? So this is your C3, and this is what? This is your C4, right? This graph is for C4 plant. Now if you see here, so the uh, amount of carbon dioxide that is present in the atmosphere, this is how much? This is 380 ppm. The optimum concentration of CO2 that is required for the growth of C4 plant is 360 ppm, right? So under normal environmental condition, the C4 plants can grow easily. They do not require anything, because the carbon dioxide that is present in the environment is higher than what is required. But if you look at the C3 plants here, the C3 plants, their optimum requirement is 450 ppm, right? But the environment have only 380 ppm. So how does this C3 plants grow? The question is this. Okay. So in order to fulfill this carbon dioxide requirement, what we do, we provide carbon dioxide from outside or from other sources to these C3 plants. Okay. So for these C3 plants, what has to be done? In order to make them grow, what can be done? So for C3 plants, Carbon dioxide has to be provided from other sources, has to be provided from other sources. And we know what is the source. We provide fertilizers, right? So this is known as fertilization effect. So after this, the plant will grow, and this is known as fertilization effect. Okay? Because the C3 plant's requirement is higher, the carbon dioxide has to be provided from other source, and this is known as fertilization effect, which means that C3 plant grow in what type of environment? So C3 plant grow in carbon dioxide rich environment. Carbon dioxide rich environment. Okay. And the example of the C3 plants are tomato, bell pepper. So these are the examples. Okay. Okay. So this is about the carbon dioxide. Now the next is temperature. Now temperature, this temperature, what it does? If you have increase in temperature or decrease in temperature, so the temperature affects enzyme, right? Temperature due to what? They affects enzymes. Okay. So whatever process that requires enzyme will be affected by temperature. Okay. Now in photosynthesis, we have two types of reaction. We know the first one is light reaction. The first type of reaction in photosynthesis is light reaction. Now, in light reaction, do we have any enzymes? Does this process use any enzyme? The answer is yes. So, light reaction, there is one enzyme known as pyridoxine NADPH reductase, right? NADPH reductase or FNR. This is being utilized for the conversion of NADP to NADPH, right? Now, in the case of dark reaction, in dark reaction, so in dark reaction is itself is known as biosynthetic phase, or it is enzymatic reaction phase. So it is called as enzymatic reaction, and a large number of enzymes are used in this process. So multiple enzymes are used. Also, we have only studied about the riboscope, but multiple enzymes are involved for each of the process. So multiple enzymes involved. So if someone asks that which of these reactions will be affected by increase in temperature or simply by temperature. So if you see, both of these reactions can be affected by the temperature, right? So we can write both light and dark reactions, and dark reaction is affected by temperature. Is affected by temperature. But which of this will be most affected? So the answer will be dark reaction. Because in the case of light reaction, we have only one enzyme. But in the case of dark reaction, it has multiple enzymes. And hence it will be most affected. So, but dark reaction will be, or you can say dark reaction is most sensitive to temperature. Be most sensitive to temperature. Okay. So this is how the temperature, this is what the temperature do. It affects the enzyme, and dark reaction is most affected. This dark reaction is most affected. Now, let's see the temperature optima. So by optima, what I mean? I mean that this is the concentration, or this is the range on which the maximum growth is obtained. Okay. So temperature optima for C3 plants, it is 20 degrees Celsius to 25 degrees Celsius. For C4 plants, it is 30 degrees Celsius to 40 degrees Celsius. And for CAM, this is 50 degrees Celsius. So these CAMs are what? Basically succulents, right? So this is 50 to 55 degrees Celsius. And suppose if we talk about hot spring algae. So hot spring, the temperature is very high, right? In that, if some algae is growing, so in that case, the temperature will be even higher. So For hot spring algae, higher temperature, right? Higher temperature. So what we are seeing here, if you see, so we know the C3 plant, they are grown on a normal temperature in a planar region. The C4 plants where they are grown in the tropical region, right? And this C5 cam plants, they are found in desert, like because these are succulents. So based on the habitat, this temperature optima is changing, right? So this temperature optima, it also depends on habitat. By habitat means where these plants grow, right? It depends on habitat. For example, tropical plants have higher temperature. This temperature optimum. Tropical plants have higher temperature optima as compared to the plant that grow in a moderate climate. Okay. So based on the requirement or based on the habitat, these plants adapt themselves. Okay. And that's why the C4 plants, those are because these C4 plants, they are where they are found on the tropical region. So they have higher temperature optimum. These camp plants, they are found in the desert mostly. So they have even the temperature optima higher than C4 plants, right? So this temperature optima depends on the habitat and the plant adapt themselves based on the habitat and hence their temperature optima increases. Now, the last factor that we'll discuss is water. Okay. So the water, we know this water is one of the reactant of the light reaction, right? Water is what? It is a reactant of light reaction, right? Hmm. Because this water splitting takes place and then the electrons are being used by the photosystem 2 and 1. Now, this water, although it's an external factor, it does not directly affect the photosynthesis. So it does not affect photosynthesis directly. Does not affect photosynthesis directly. Okay. But what happens if there is yes the decrease in S2O concentration? So say decrease I'm showing by this. So two things can happen. Okay. The first thing, if there is decrease in S2O concentration, leaf wilting. So your leaf it will wilt, right? So leaf wilting will take place. Leaf wilting, or you can say leaf become flaccid. Okay. And what happens if there's leaf wilting? So there will be decrease in surface area because suppose their leaf is first this, it has this much of surface area. Now it has wilted to this. So the surface area has decreased, right? So what is happening? There is decrease in surface area. Decrease in surface area. And this decrease in surface area. Now it has decreased in surface area. There will be less absorption of light, there will be less absorption of carbon dioxide, and that will ultimately leads to decrease in rate of photosynthesis. It will lead to a decrease in rate of photosynthesis. Right? So this is the first thing. Now, when there is less water, low water concentration, the stomata will be closed. So it will lead to closed stomata. Now, if there is less stomata will be closed, there will be decreased CO2 concentration because less amount of CO2 will enter. So there will be decreased CO2 concentration. And this decrease in turn lead to decreased photosynthesis rate. Okay. So these are the four factors that affect the rate of photosynthesis. Now with this, we have completed this chapter. Let's solve few questions that is given in the NCRT. So the first thing is, and it is kind of revision for you as well. So the first thing is the cell types in which the Calvin cycle takes place. So we have to answer in terms of C3 plants and C4 plants. So in C3 plants, where the Calvin cycle takes place, Calvin cycle is what? It is dark reaction, right? Or C3 cycle. So C3 cycle in the case of C3 plants, where it takes place in the mesophils, right? Now, in the case of C4 plants, where the C3 cycle takes place, it takes place in, so if you remember, in mesophil plants, we have two types of cells, right? Here is a mesophil, and then your bundle sheath cells, right? Now, the C3 cycle, it takes place in the chloroplast that is present in the bundle sheath cells. So CO2 is taken here, but it is getting ultimately converted, and then it reaches to the chloroplast of bundle sheath cells, where the C3 cycle takes place. So the C3 cycle in the case of bundle C4 plants, it takes place in bundle sheath cells. Bundle sheath cells. Now, what cell type in which the initial carboxylation reaction occurs? So in the case of C3 plant, it has only one type of cell, that is mesophil cells. So in C3, it will happen in mesophils. Now, in the case of uh, this C4 plants, where the initial carboxylation takes place, so the carboxylation takes place here in the mesophil itself, right? Because here CO2, it binds to PEP, right? So here, in the case of C4 also, it takes place in the mesophil cells. Now, the third question is, how many cell types does a leaf have that fix CO2? So, how many type of cells are required for the fixation of CO2 or for the dark reaction? So, in C3 plant, like I said, it has only one type of cell, that is mesophil. So there is one type. But in the case of C4 plant, we have two type of cells, right? We have two types, and that is what? This bundle sheath cell and mesophil cells. Okay, so now the next question is, which is the primary carbon dioxide acceptor in the case of C3 and C4? So if you remember, in the case of C3, we had what? We had RUBP, right? In the case of C3, the primary carbon dioxide acceptor is RUBP, while in the case of C4, we can see here the primary acceptor is PEP. So when carbon dioxide reaches the cell, RUBP accepted or RUBP undergo carboxylation, while in the case of C4 plants, it is the PEP which undergo carboxylation. Okay, the next question is number of carbons in the primary carbon dioxide acceptor. Okay, so the number of carbons that is present in primary carbon dioxide acceptor. So the primary carbon dioxide acceptor in the case of C3 plant is RUBP, and the number of carbons that are present in this is 5. Okay, and in the case of PEP is what? It is 3 carbon molecule. So in C4 plant it is 3. Okay, now next is, which is the primary carbon dioxide fixation product? So by this, what it means that when carbon dioxide comes, it binds to this primary acceptor, right? That in the case of C3 is RUBP, right? And in the case of C4, it is what? CO2 plus PEP, right? Now, this CO2 plus RUBP, what it forms? So, what it forms? It forms two molecules of phosphoglyceric acid. It forms two molecules of 3-PGA, right? And this, what does it forms? It forms one molecule of oxaloacetic acid, right? So, which is the primary CO2 fixation product? So, the first product or the primary product is what? In this case, it is PGA, and in this case, it is oxaloacetic acid. That is OAA. Okay. Okay. Now, the next is number of carbons in the primary carbon dioxide fixation product. So the first product that is formed, how many carbon molecules do we have in that product? So in the PGA, how many carbon molecules do we have? So the PGA or phosphoglyceric acid, it is 3 carbon molecule. While this oxaloacetic acid, this is what? This is 4 carbon molecule. Okay. Now, the next question is, does the plant have rubisco? So we know this rubisco is universal. It is present in all type of plants. So C3, C4, CAM, in all type of plants, this rubisco is present because this rubisco ultimately leads to the Kelvin cycle. And Kelvin cycle is a universal cycle. So both of these plants have rubisco. That is true. Now, does the plant have pepcase? So pepcase is pep carboxylase. It is not present in the C3 cycle, right? Because in C3 cycle, the rubisco initiate the process. But this pepcase is present in the C4 plants and is also present in CAM plants, right? Okay. Now the next is which cells in the plant have rubisco? Now we have said that rubisco is present in both C3 and C4 plants, but in what kind of cells? So in C3 plants, because it involves only one type of cell, so rubisco is present in mesophils. But in the case of C4 plants, where the rubisco is present, so what I said that here, if you see, the CO2 enters in the mesophil cells, but they ultimately gets converted into oxaloacetic acid, and then ultimately the carbon dioxide here it enters into the bundle sheath of the chloroplast of the bundle sheath, right? And this chloroplast is rubisco. So in the case of C4 plant, the rubisco is in bundle sheath cells. In C4 plant, the mesophils have pepkes and bundle sheath cells have rubisco. Okay. Now, the next question is carbon dioxide fixation under high light condition. So, if you see C3 plant, this C3 plant have low temperature optima, right? And if temperature is low, it means low temperature optima. It means that it will work better in low light intensity, right? So, if you try to fix carbon dioxide under high light condition, the C3 plant they will have low carbon dioxide fixation. While the C4 plants, because they have high temperature optima, so even the high light intensity, and also they are present in the tropical region where the light intensity is high, so they are adapted, right? So the carbon dioxide fixation under high light condition will be high in the case of C4 plants. Now the next point is whether photorespiration is present as at low light intensities. So when does photorespiration occur? So photorespiration occur when rubisco binds to O2, right? It occurs when rubisco, this enzyme, it binds to oxygen. But we know that in case of C4 plants, rubisco do not get any oxygen to bind, right? So even if light intensity is low, high, this C4 plant will never form the or undergo photorespiration. Here it will be negligible. Now the question is for the C3 plants. So C3 plants under low light intensity, will it have photorespiration? The answer is no, because here what happens? Here at under low light intensity, your carbon dioxide fixation is taking place, and this reverse will bind to carbon dioxide, right? Now, what will happen if we have high light intensity? In this case, if there is high light intensity, because these three C3 plants are not adapted to high light intensity, they will undergo photorespiration. So here, the photorespiration will be high, while the C4 plant still it will be negligible. Okay, now this is C3, C4. Okay, now the next is whether photorespiration would be present at low carbon dioxide concentration. So if the carbon dioxide concentration is low, in this case, the photorespiration will happen in the C3 plants because if there is low CO2, the ribosco will bind to oxygen, right? But in the case of C4 plants, even at the low oxygen carbon dioxide concentration, we know that C4 plants have the mechanism to concentrate or increase the concentration of carbon dioxide. So even the carbon dioxide is present in the low concentration externally, the cells will take them and they will increase their concentration inside the cell. Hence, in the C4 plants, there will be no photorespiration. So it will be negligible. It will be negligible. Okay. Now, next is whether photorespiration will be present at high CO2 concentration. When there is high CO2 concentration, the reverse will bind to CO2 only, and hence no photorespiration will happen. So the answer will be negligible. The answer will be negligible in both the cases. Now, if we talk about the temperature optima, we have just seen the temperature optima for C3 plant is for 20 to 25 degrees Celsius, and C4 plant is 30 to 40 degrees Celsius, right? Now, finally, let's talk about the example of C3 plant. So the C3 plant example is a rice, wheat, cereals, and in the case of tomato that we saw, and in the case of C4 plant, it is sugarcane, sorghum, maize. So these examples are important. This is maize. Okay. So this is the complete revision of the photosynthesis where these questions. And with this, we have completed this chapter. That's all for today. Thank you.